English teachers पढ़ाए तो simple लगे everything. Leaderboards and quizzes से
Amazing.
learning हो जाए happening. समझ आएगा तो मजा आएगा.
Vedantu, learn live online. To attend a free live class, download the app now.
Hey students, good evening, namaste and welcome to part 2 of our class 10 ICSE Biology Specimen paper, the latest one for the academic year 2019-20, which means it's aimed at giving you practice for your board exam, your ICSE board exam 2020. Okay, so as you know, in the previous session, we are already done with the discussion of section 1 of the paper. And you know what? Section 2 actually has questions worth 40 marks, just like section 1. But again, it seems a little more complex in terms of the kind of questions that are asked. Okay, so just to make it easier for you and for myself, in fact more for you so that you would be able to focus better, I decided to break down section 2 also into 2 parts. So totally we are going to be having 3 parts exclusively for specimen paper discussion. One that we had yesterday, one that's happening now, which will be discussing part of section 2 of the paper, and the last part will be happening tomorrow. Okay, and I hope your new year is all absolutely going on fine and great. I hope all of you have started the wonderful, amazing year 2020 with a lot of positivity and newer resolutions. Let's just consider this as a good time of the year to become a better person. Let's correct ourselves from whatever we have learned in the past year 2019, and let's try not to repeat the same mistakes again, and let's just try and strive to be a better person every single day, right? I'm sure all of us are capable of that. The only thing we have to do is get rid of all laziness, get rid of all negative thoughts. Of course, it's easier said than done. I am also talking about myself, of course, not just about you. All of us have to learn a lot of things about ourselves. So let's just aim at becoming a better person, as simple as that. Okay, and with that, let's get started. As you know, here at Vedantu, we have launched a lot of amazing courses just to help you achieve much better, just to help you ensure that you achieve the best of your potential and reach the maximum extent of success, especially now as far as your board exams are concerned. We are always here for your support, not just on YouTube, but on our regular Vedantu.com wave platform also, wherein we have launched a lot of courses, among which the micro courses have been discussed about a lot, because these are exclusively aimed at those of you who want specific chapters or specific subjects help. Okay, so because everyone's strengths and weaknesses may not be the same. You may be good at math, but you may be bad in bio. But you may be good in bio, or you may be bad in physics, something like that. And even within a particular subject, you may have a chapter which you're really good at. You may have another chapter that you really suck at. So whatever it is, keeping your strengths and weaknesses in mind. And I hope this is also a way of getting you to identify your own strengths and weaknesses, because we are just barely a couple of months away from the board exams. So start identifying your strong and weak points right now, and do get enrolled in our micro courses accordingly. And it's very, very simple. And the best part is you get all of these features, all of these amazing features you do not get on our YouTube sessions. You get more personalized sessions, because you get to interact with the teacher a little more, just like a normal classroom. That is basically why these courses are certainly beneficial. Okay, and the best part is they are very reasonably priced, starting as low as ₹11. All you have to do is make a note of this link. Do click on the link. Do visit the micro course section and use this code AMMIC to avail a great discount. Here I again have a huge surprise for you now, because we are on a weekend. I have to tell you this. This is something that we have valid only for Saturday and Sunday, only for the weekend, an exclusive weekend special offer, wherein, yes, we are actually and literally trying to flash happiness, and the offer ticks only between 7 p.m. to 9 p.m. What exactly am I talking about? Well, yes, we have unlocked all our micro courses at flat rupees 11. Yes, you heard it right. So make use of this offer, make use of this opportunity, my dear children, because great quality content at such a reasonable price. Yes, so apart from this, we also have what is called the crash course, and for ICSE Class 10, we have crash courses designed for all the chapters across the subjects physics, chemistry, bio and math. Basically your science and math subjects, because these are usually the subjects which most children ask for help. Okay, so we basically have live classes by some of the best teachers across the country, some of the best teachers of the country. You have access to a lot of comprehensive tests, assignments and detailed analysis of those. You get to discuss your doubts with the expert academic mentors out here at Vedantu, and you also have an amazing feature of having the replay or recording of those classes that you have missed. And yes, important tips and tricks on how you can crack your board exams effectively, rank booster quizzes, past paper analysis and so on. And all of this again available at a very attractive price of 7,999 only. So just to avail it at this attractive price, imagine all your subjects, the entire year's syllabus for these 4 subjects, okay, at such a reasonable price. All you have to do is follow these instructions, 3-step instructions. Do go visit this link and you can follow the instructions, and you can use the code AMCC to get this discount. Okay, so do make use of this opportunity. This is for those of you who just want some extra bit of guidance, who just want that, I would call it a winning edge, that final brush up or final sharpening of your skills and knowledge. Yes, just before your board exams. And as you know, Vedantu's learning app has been awarded the User's Choice App Award 2019. Thank you so much for being a constant source of support. I really hope you continue using the app, because we keep updating it regularly. We keep updating and upgrading on the quality content out there. Our team here works very hard for that. And yes, do stay in touch with us through our Telegram group. A lot of our teachers would be able to answer your questions related to academics out there on the Telegram group. Yes, the link is provided down there in the description box below. So do have a look, click it and join us. And yes, guys, do hit the like button if you have been enjoying all our ICSE series sessions. Hit the like button right now, please. And as I told you yesterday, I would really and sincerely request you to share this video with all your ICSE Class 10 friends, because it would certainly benefit them, because it's the specimen paper discussion. And do subscribe to the channel Vedantu 9 and 10 if you haven't done it yet. Yes, and with that, let's get started. Today, as I told you, we shall be discussing part of section 2. Okay, the first half of section 2, which is totally for 40 marks. So basically, you will be given 6 questions, among which you can attempt any 4. Okay, but the tricky thing is you have to be able to wisely choose, because even if they say 6 questions from which you have to choose 4, it sounds like a very simple task. Okay, choosing 4 out of 6 is no big deal, but then the number of sub questions each of them has is like a lot. So you have to first go through the paper entirely during those few minutes before your exam starts, that you're given exclusively to read the question paper. Make sure at that point of time you read through everything, so that you are able to maximize your ability to score full marks in a particular question, or maximum marks in a particular question. Okay, I hope you understand what I'm trying to say. Anyway, we'll discuss all such tips and tricks in another exclusive session, but today let's get started with the first question of section 2 of your specimen paper, biology. Okay, yes, as you can see, we are given a diagram. So study the diagram given below and answer the questions that follow. Okay, so there are questions which will totally add up to 5 marks based on this diagram. Okay, so I understand the diagram is not very clear, but with a little bit of zooming in, you will be able to see what is being explained or what is being illustrated in the diagram. Yes, it is the blood vessel, the blood cells inside the blood vessel. Okay, that is what you see in the very first one. The first question is, name the cells labeled 1, 2 and 3. So basically 1, 2 and 3. What are they? Of course, they are your formed elements of the blood. 1 is certainly, from the shape you can make out, although it's not colored, 1 is RBC. What is 2? It's amoeboid in shape, so obviously it has to be a WBC. And C or 3 is something that's differently shaped from these two, but it is a formed element in your blood, so it has to be the platelets or your thrombocytes. Okay, so RBC, WBC and platelets. Yes, there goes our answer. 1 RBC, 2 WBC and C platelets. Okay, yes. Okay, so let's go ahead to the next question based on this diagram, which is, identify the phenomenon occurring in A and explain the phenomenon. Okay, so what exactly is the phenomenon that's occurring in A? Of course, in A, you can just see the blood vessel and you can see some formed elements, right? But if you closely observe it, you can see that the white blood cell, right? Number 2, the element number 2 that we named as the white blood cell is actually doing something. What is it trying to do? It's trying to squeeze itself out through pores on the walls of the blood vessels. Right? So there is a name that we give to that process, right? We call it diapedesis. Okay, this process is called diapedesis, and what exactly is it? It's basically the process by which blood cells, specifically white blood cells, pass out through tight blood capillaries. We are talking about this particular scenario. Okay, white blood cells are trying to squeeze out or pass out through the intact capillary walls into the surrounding tissues. Why do they do this? When there is an injury or inflammation that occurs, they try to do this. They try to rush to the site of injury. Okay, so that is called diapedesis. Yes, so that gives you another 1 mark. And the next question. Mention 2 structural differences between 1 and 2. Okay, 1 and 2, which means basically the RBC and the WBC, 2 structural differences. How would you differentiate between them? Yes, children, tell me. Structural differences, not any random difference. Yes, functionally, of course, we know the RBC does a certain function, which is carry around oxygen because of hemoglobin, and white blood cells basically help in fighting infections, right? But structural differences are specified in the question. So how would you write this? Yes, RBCs are biconcave in shape. What do I mean by biconcave? First of all, yes, biconcave means something like this. Okay, biconcave in shape, whereas WBCs are amoeboid in shape, no specific shape. Yes, no specific shape, they are just amoeboid. And that is because they have to phagocytose, or they have to help in fighting infections, engulf the bacteria or other foreign bodies and fight infections. That is why we describe them as amoeboid in shape. Basically, shapeless structures are described as amoeboid. Okay, so that's about a structural difference between the two. Now going ahead, question number 4 here. Name the process occurring in B and C. Okay, now B and C, if you closely look at it, what is happening in B and C? This is B and this is C. Now you can see that the white blood cell, right, there's an arrow here. This white blood cell has squeezed out itself, right, squeezed itself out through the capillary wall. It's come out. What is it doing? It's basically trying to engulf bacteria and In C, you can see it's already engulfed it and it's trying to digest and get rid of the bacteria by killing it using its own enzymes. Okay, what do you call that process? I've actually indirectly told you the answer just a few seconds ago, or probably a minute ago. We call that process phagocytosis. Just like how amoeba gets itself food. Phagocytosis. That's basically what our WBCs do when they have to attack a foreign material. Okay? Yes. Now we have one more sub-question. State the importance of this process in the human body. Why do you think phagocytosis has to occur in the human body? Very, very simple, right? Of course, phagocytosis is the means of fighting infections or getting rid of foreign bodies from the body, from the human body. Basically, it's a defense mechanism, right, for the body to fight against infections. As simple as that. And 5 marks right there in your kitty. Okay? And question number 2. Again, sub-question number 2 under the main question 2. Okay? Give one difference between each of the following pairs on the basis of what is given in the brackets. Wow. Okay, you are supposed to differentiate between the pair of words or pair of terms that's given to you. Not just any random difference. You will be specified based on which point you have to write the difference. Okay, so you have to know that very, very, very specifically. Smart ICSE people. Mitral valve and aortic semilunar valve. You are supposed to differentiate between the location of the mitral valve and the location of the aortic semilunar valve. So yes, where have we learnt it before? Yes, the human heart, of course, but where in the human heart? Yes, so for your reference and to make things easier for you, I decided to put an image with exclusively the valves marked out in the human heart. We are talking about the aortic semilunar valve and the mitral valve. But where is the mitral valve in this? Yes, guys? Yes, mitral valve is also known as the bicuspid valve. So, tell me the difference between the location of that and the location of this. Where do you find this and where do you find that? Yes. Now, for this, you must very clearly know which the right and left sides of the human heart are. I have said this very clearly in my session. I remember that. Yes. Now, imagine this is my heart because I'm standing in front of you and this heart is right there corresponding to the direction in which my heart is also oriented. So this is right and this is left. So considering that, where do you think the mitral valve is or the bicuspid valve is? It would be between the left auricle and the left ventricle. Correct? The left auricle and the left ventricle, and between them, the bicuspid valve. Yes? What about the aortic semilunar valve? You find it right here at the junction between or at the start of the aorta, opening of the aorta at the left ventricle. So how would you write the answer? Yes, mitral valve or the bicuspid valve is between left auricle and ventricle, while the aortic semilunar valve is at the point of origin of the left ventricle and aorta, which means at the point of contact between the left ventricle and the aorta, basically point of origin of the aorta at the left ventricle. As I said very, very clearly, you must know the difference between right and left sides of the human heart. Okay? It's not when you're writing it in your paper, imagine that you're drawing the heart of a patient who's lying in front of you and you are the surgeon. So what would be the patient's right and what would be your right? Opposite, right? Correct? Yes, so many rights and corrects. Anyway, be clear about that and then you will never go wrong in this concept. Okay? Yes. And with that, let's go to the next difference between, yes, now you're supposed to differentiate between something very, very simple, hydrotropism and thigmotropism based on stimulus. What exactly are they? They are basically different types of tropic movements, directional movements. Hydro tells you the answer. The stimulus is exactly, it's water. And thigmo, T. T for touch. Yes, so this is your answer. Hydrotropism and thigmotropism. And this is how you can write your answer. Now children, these pictures, I've just put it out there for your reference. I've put pictures only for those questions for which I thought that pictures could add to your way of understanding. Okay, for very, very simple concepts, I've not put any images. Remember, just because I've put images here doesn't mean that you are expected to draw these images for such questions. Okay, that's not what I mean. You just have to write the answer crisp and to the point. Okay, so that will be your answer to this question. Next one. Yes, now it's metaphase and anaphase, which is based on the position of chromatids. Yes, where do you talk about metaphase and anaphase? They are definitely phases of cell division. Have a look at this for your reference, metaphase and anaphase. So with this, you will certainly be able to say the difference between the position of chromatids in either case. Metaphase, yes. Meta is middle. So that means the chromatids are arranged in the middle of the cell or along the equatorial plate of the cell. What about anaphase? Ana means opposite. So what does that mean? The chromatids are at opposite poles of the cell, and that will be your answer. Yes? And now sub-question number 4. Demography and population density. Okay. This is not very hard either. It's only the definition that's been asked. Demography, if you remember, because it was one of the last chapters that we discussed in the last couple of weeks, I'm assuming you would be able to recall what it is. Demography is basically the statistical study of human population with reference to size and density, distribution, and various other vital statistics. Whereas population density is the number of individuals per unit area or per square kilometer, to be specific, at any given time. Okay? That is basically the difference between these two. Demography is basically a study. Population density is the density of the population. Okay? Yes. Now the last difference between question is turgid cell and plasmolized cell. Yes, and that has to be based on tonicity of the surrounding solution. Okay, so what are we talking about? If you have understood at least something from the class, you would remember we are talking with reference to osmosis, or we are talking with reference to tonicity. What is hypertonic, what is hypotonic, what is isotonic. Yes, and I've put this picture out there for your reference. If you placed a cell in a dilute solution or a hypotonic solution, water would enter into a cell. I'm talking about a plant cell specifically. Okay, water would enter into the cell and it appears turgid. Okay, whereas in an isotonic solution, nothing happens. There is inflow and outflow pretty much equally. Okay, you call that flaccid. And we also have to know what plasmolized cell is. Now, plasmolized is with reference to only a plant cell, which is why I put the image of only a plant cell here. Plasmolysis is basically the process by which the cell contents shrink away from the cell wall because it is placed in a very, very highly concentrated or hypertonic solution. Yes, because when it's placed in a hypertonic solution, water flows out from the cell into the surrounding solution. So, plasmolysis or plasmolysis happens. You describe the cell as a plasmolized cell. Okay, so how do you differentiate? To the point. Turgid cell is a cell that appears turgid when placed in a hypotonic or dilute solution, case 1. This is just there for your reference, so do not worry about that for now. Plasmolized cell is a cell which becomes so, or a cell which becomes plasmolized when placed in a hypertonic or concentrated solution. Okay, just apply a little bit of basic logic and you will be able to understand what is happening. Okay, yes, and with that we come to the end of that question number 2. The main question number 2 is done. Okay, now an outline sketch of a tree is shown in the diagram below. Study the same and answer the questions that follow. Okay, again a not so great image, but you can certainly understand what's being asked in it. A, B and C are labeled near the tree. You can see what A is, you can see what B is, and you can see C, right? Name the phenomenon that's labeled A. What do you think is happening in A? You can see arrows out there. What is happening? Yes, it has to be transpiration. Yes, the process by which water is lost in the vapor form from the inside of the plant to the atmosphere. And that is actually your second question. Because I was too excited, I just explained it to you. Second question is explain the phenomenon occurring in A. Basically explain the process of transpiration. So this is all that you have to write because it's a 1 mark question, right? Now, question number 3. What is the importance of this phenomenon in plants? Tell me. Why does transpiration occur? Yes, one is to cool down the plant body, and secondly, to help in distribution of water and minerals from the roots to upper parts of the body. Okay, yes, so you have to list down those, and again you get a half plus half, 1 mark, completely yours. Very, very simple, right? Okay, now sub question number 4. Explain the role of any 3 external factors that will increase the rate of photosynthesis. Okay, so let's now look at sub-question number 4 of this, which asks, explain the role of any 3 external factors that will increase the rate of this phenomenon. Now, if you remember what we discussed in section 1, there was a question that was similar, that was somewhat based on this concept. So, keeping that in mind in the multiple choice section, okay? So, keeping that in mind, let's see how many of you remember the factors that affect the rate of photosynthesis. Okay, have a look at it. This is just based on what we discussed in the class. Roughly an image that describes transpiration, and to mention a factor that increases transpiration, one is intensity of light. Yes, greater the intensity of light, greater would be the rate of transpiration. Okay, secondly, there is another factor which is also external, and that is temperature. Remember, we discussed hot, dry and windy. Yes, that's what I said. You will get a hint from that. Increase in temperature increases the rate of transpiration. And wind velocity, we've discussed this also. Greater the velocity of wind, greater would be the rate of transpiration. Okay, and if you write those 3, you get full marks for that. Now, what do the direction of arrows in B and C indicate? B, if you can see, this is B and this is C. But where are the arrows? This is the arrow for B and this is the arrow for C. C shows uptake of something from the soil. B shows movement of something up the plant body. What do you call that? There are names that we use, right? Yes, B is called ascent of sap. Remember? Yes, and C indicates absorption of water from the roots, from the soil by the roots. Okay, so that is what the arrows B and C indicate. Again, very, very easily you have scored 5 full marks. Yes, now we come to the next sub-question of question 3. Mention the exact function of the following structures. Okay, the first one being iris. Which part of your body do you find iris in? Yes, it's part of your eye. But what does it do? Yes, look at this image. This is your iris, and its basic function is to control the diameter of the pupil of your eye so that it can control the amount of light entering into the eye. For example, it shrinks the pupil. You can see that, right? The blue structure is what we are talking about as the iris. It shrinks the pupil when there is too much light and widens it when there is not enough light. Okay, so that is the function of the iris. The next one, plasma membrane. Very, very simple. All of us have been hearing from grade 5 or 6 about what is plasma membrane, right? It's the outermost membrane of an animal cell and the second to last or second to the outermost membrane in a plant cell, right? What does it do? It basically is a semi-permeable membrane that helps in osmosis in plants. Okay, in animals also it's something very similar to this. Selectively allows particles to enter and leave the cell. That is the plasma membrane. Now nephron. Yes, what is the nephron? As long as you don't get confused between nephron and neuron, it's all right. Nephron is basically the smallest or the basic structural and functional unit of your kidneys. What does it do? Remember that complex process that we discussed. Mechanism of urine formation. Yes. So what is the function of the nephron? It is urine formation. It performs the function of urine formation. Okay. So that is also done. The next one, thylakoids. Where does it strike bells? Where have you heard this term? Is it in a plant or is it in an animal? Yes. Put in all your comments in the chats. I will be reading them when the premiere is happening. Yes, guys, it is undoubtedly in a plant cell. And where exactly in a plant? This is the structure of a chloroplast, which is a part of the plant cell. A simple organelle of the plant cell inside which you find the thylakoids. But what does it do? What is the function of the thylakoid? Thylakoid is basically this. There's one single green structure that you see is the thylakoid, and a stack of thylakoids is called a granum. What does it do? It contains chlorophyll and hence it helps in absorption of sunlight for photosynthesis, basically helps in the light reaction. Okay? Yes, one more to describe the function for which is the hydatodes. Yes, again, where do you think it is? In our body or in an animal's body? I mean, we are also animals, but in another animal's body or in a plant body? Yes, it is in a plant body. This is where you would find a hydatode. What is it involved in? It's involved in a process called guttation. They discharge water from the interior of a leaf to the leaf margins, as you can see here, right? By a process called guttation, which is similar to transpiration, but in transpiration, it's lost as vapor form. Guttation, it's lost as water itself, water droplets. Okay, that is what hydatode is involved in. This is basically the structure of one single hydatode. Okay, all these associated apparatus. It's just there for your reference, so do not worry looking at the complexity of that. Okay, now going ahead. Yes, we have come to question number 4. Okay, what do we have to answer here? It looks a little complex, but it is not. Trust me. Given below is a diagrammatic representation of a defect in the human eye, which has been corrected using a suitable lens. So study the diagram and answer the questions that follow. If you answer all of them correctly, you get 5 full marks. Okay, the first one is identify the defect that's been corrected. What do you think it is? Look at what kind of lens is provided. Yes, it said it's been corrected using a suitable lens, right? Look at what kind of lens is provided to correct it. It is a convex lens. So, tell me the answer. What sort of a defect is it? Yes, it is called hypermetropia. Okay, so this is just there for your reference again. This is what the hypermetropic eye looks like because it is simply called long-sightedness or far-sightedness, wherein the image, instead of being focused on the retina, gets formed behind it. And because of that, nearby objects cannot be seen very clearly. Yes, as you can see in A and B, here it's a nearer object which cannot be seen clearly because the image is formed behind the retina. Whereas in B, It's a farther image, okay, which can be properly focused. But in the third one, you can see the correction that's given for the hypermetropic eye using a convex lens. And because of that, whether near or far, all objects can be seen clearly, okay. So the condition or the defect is called hypermetropia or far-sightedness or long-sightedness, okay. Now, next one, mention 2 reasons for the above defect. Why does hypermetropia occur? It can be because of low converging power of the eye lens, because of weak action of the ciliary muscles of the eye, okay. And secondly, abnormal shape of the cornea, the outermost covering, can also be a reason for hypermetropia, okay. So these are 2 possible reasons for this. Now, third question based on this, label the parts numbered 1 to 4, basically 1, 2, 3 and 4. Yes, tell me very quickly, what is 1? 1 is basically the convex lens or the spectacle lens, right, which is given to correct the condition. Secondly, 2 is the lens of the eye of the affected patient. What is 3? 3 is basically this outer part, which has to be the cornea. And what is 4? The point where the image is formed. What could that be? Yes, it has to be your retina. So, here are your answers. A slightly clearer image as compared to the previous one. 1, 2, 3 and 4 as we have discussed. Okay, children? Alright. And the next question, state the functions of the parts numbered 3 and 4. Basically, you have to write the function of the cornea and the retina. Yes, what does the cornea do? Cornea is the transparent front part of the eye, which covers the iris, the pupil and the anterior chamber of your eye. And the retina, we've indirectly told or mentioned the function of it. It receives light which the lens has focused and converts the light into neural signals, and it can send these signals on to the brain for visual recognition. Basically, it's the place where image can be formed in your eye because of the coordination between the retina and your brain. Okay? Yes. One more. Last question based on this image. What maintains the shape of the eyeball? Not exactly or directly related to this image, but related to the eye. Okay? Yes. What maintains the shape of the eyeball? It is something called the vitreous humor. This basic gelatinous filling that's out there inside this, which maintains the shape of the eyeball. Okay, now do not worry, children. I've already told you this yesterday. I understand that many of you want help with the topic sense organs, which are the structure of the eye and the structure of the ear. I have taken it very seriously into consideration. You can expect a session on that within the next few days. Okay, so even if you have not understood this crystal clearly, never mind. This is just there for your understanding of the specimen paper. Okay, no worries. Now, the last question for today, which is a set of 10 name the following. Not name the following exactly. It says biological or technical name for the following. 5 marks, 10 questions. Very, very quickly, who is going to give me the answer? Okay, let's see. The quick actions which are involuntary and controlled by the spinal cord. What are they? Yes, reflex actions. Yes. Second one, the structure formed after the release of ovum from the Graafian follicle. Seems a little complex, so I have put an image for you there. After ovulation, when the ovum matures, when the egg is released, once the ovum is released, there is something that is left off from the follicle. That is called the corpus luteum. So that becomes your answer, corpus luteum. Okay, once again, look at the question. The structure formed after the release of the ovum from the Graafian follicle. Okay, the leftover after the mature ovum is released. Okay, corpus luteum. The surgical technique used in human females to prevent pregnancy. Yes, a very, very effective way of contraception, which is called tubectomy, wherein the fallopian tubes are cut and tied. Okay, and going ahead to the next one, the stage of cell division in which the nuclear membrane disappears and the chromosomes become short and thick. Let's see how many of you remember the stages of cell division. Yes, prophase, metaphase, anaphase, telophase. Among these, where is it that the nuclear membrane disappears? Which was there disappears. I'm giving you a very, very good hint. When it disappears, it's normally represented as dotted lines. It's there in prophase and it's there in telophase. But using your logic and your basic understanding, think about where it's disappearing and where it's reappearing. Yes, in prophase, it has to be disappearing and the chromosomes becoming condensed and thickening. So your answer there is prophase. Telophase is where the nuclear membrane reappears after division or separation of the chromatids. Okay, so the answer is prophase. Now coming to question number 6. The canal through which testes descend into the scrotum just before birth in a human male baby. Yes, how many of you remember this? The testes descend into the scrotum through a canal, which is called by a name. It is called the, yes, inguinal canal. Okay, for your reference, I've put this there, the testicle and the scrotum, the sac-like structure into which the testicles would descend. Okay, an inguinal canal would be the canal through which this descending happens just before the male baby is born. Okay, all right. Next one, question number 7. Repeating components of each DNA strand lengthwise. Again, going back to one of our first chapters, structure of DNA. DNA is deoxyribonucleic acid. If you recall, nucleic acids are polymers of something that we call, yes, nucleotides. As you can see here, one nitrogenous base along with a sugar or a deoxyribose sugar and a phosphate forms a nucleotide. Repeating units of nucleotides form a DNA. Okay, so nucleotide will be your answer for question number 7. Question number 8 is extremely simple. Site of photosynthesis in a plant cell. Who am I asking this to? Yes, all of you are smart. I know you have said the answer is chloroplast. Yes, the constituents which cause the natural quality of the environment to deteriorate. What is it? It's been described in a seemingly complex way, but it's something very simple. Bangalore is full of this, Delhi is full of this, Chennai also has lots of this, Bombay also has lots of this, Kolkata also has lots of this. What is it? Yes, pollutant. Pollutants are the constituents which degrade the quality of the environment. Next one, the last one for today's session. Okay, the defect of vision in which some parts of the objects are seen in focus while the others are blurred. Okay, hypermetropia, we've seen it already. It's farsightedness, so that's not this case. Opposite of that is a case called short-sightedness, which is myopia. So that's also not the case. So what is this? Some parts are seen in focus while others seem blurred. It's a different condition. Yes, look at this. This is a normal eye, and when the lens is irregular, this blurring, irregular blurring happens, and we call that astigmatism. So astigmatism is your answer to the last question of today's session. So remember, children, we are not done with the entire paper yet. The rest of the paper will be discussed. It will take us another hour to discuss the rest of the paper because, as I told you, it's 4 questions out of 6, but because it is meant to help you out, we have to discuss all the 6 questions, right? So I will be taking another session for you tomorrow on the remaining parts of the specimen paper. So stay tuned. Do not miss that session. If you have enjoyed today's sessions, if you have found the session useful, do remember to hit the like button. Right now, I thoroughly enjoyed doing the session for you, although I would have loved it better live, but my respiratory conditions did not allow me to do that. And do share it with all your ICSE class 10 friends, children, because all of them will certainly benefit from this. It is a great way of boosting your confidence, solving the latest specimen paper. And yes, subscribe to our channel if you have not done yet, Vedantu 9 and 10. And just in case you want any help from my side, remember to get in touch with me at my email, ambika.gopalakrishnan@vedantu.com. And until we meet again in part 3 of ICSE paper discussion tomorrow, this is Ambika signing off. Bye-bye.